much. I am just so delighted to be here and give a keynote here at this international conference. It's really delightful for me to get to share my journey through science with all of you. And I wish I had had this kind of community and opportunity to connect with other students who were also interested in computational biology and bioinformatics when I was in junior high and high school. Although I think I didn't have a nuanced understanding of what the field was at that time. So I'm just really excited for all of you and thankful to be a part of this experience with you. So what I wanted to do today was talk to you a little bit about my journey as a scientist, how I started in this field, and the questions that still excite me today. And also to tell you about, as sort of a vignette, one research project that is a kind of data-driven research project in my group that we had one large publication on a few years ago in collaboration with the company 23andMe Incorporated, and that we continue to work on. But when I was reflecting, as I was thinking about writing this talk, I started thinking a lot about my graduate advisor here, Marcus Feldman. He just turned 80 on this past Monday. This is a picture from now a little while ago with him and my oldest daughter. I have 3 children. So this is my oldest when she was 3, and it will be turning 10 in January. So Mark has been a really important person in my life, and what's made me the scientist that I am today is trusting in something that he told me when I was finishing my PhD with him. I was trying to decide on what research appointment to take next. I was doing what's called a postdoctoral researcher appointment before I became a faculty member. And I had a couple of choices, and I felt very strongly like there was a right answer and a wrong answer, and I just didn't know what decision to make. I was really conflicted about it. And at one point, he and I were having breakfast together, and he was trying to help me decide and had asked me to meet him on a Saturday morning, much like today. For me, it's Saturday afternoon. And I said, will you just tell me what to choose? And he said, no, I'm not going to tell you. I don't want to influence you. But he said something to me that has stayed with me since. He said, when you look back on your career, you'll realize that the events that most influenced you were accidents you never could have anticipated. And so he said to me, it feels like there's a right and a wrong answer right now, but the truth is that this choice isn't a big choice. And in fact, there are other things you'll never be able to anticipate that will change your career. And he was right. So I think a lot about those accidents, and I want to share some of them with you now. So going back to a stage of life where I was, like many of you in this room, I actually started working with Mark as a high school student. I was interested in participating in science fairs, and I had contacted a lot of different professors sort of randomly. It so happened that my older sister knew Mark from college and suggested that I talk to him. And so I ended up working in his lab for a summer as a high school student. And this is what he and I looked like back then. I was 15. And I ended up entering a competition called the Westinghouse Science Talent Search then. It's now called the Regeneron Science Talent Search. I also participated in the International Science and Engineering Fair, which may be something that many of you are thinking about or have heard about. At that time, Mark's lab had a number of students. It was quite large. And he has a history of mentoring people who've gone on to careers in academia. So these were some of the people in the lab at the time, and they're all professors now at a range of institutions in the United States and also in New South Wales, and many of them have worked internationally. So being around that group really influenced me, and especially the fact that Mark had set the tone that a high school student working in the lab should be treated just like a graduate student, just like a colleague. So all of these people were incredibly kind to me. So what did I work on? My project was a bioinformatics project, but I had no computer science background. I had just studied, math and biology, and I was interested in them. My project involved implementing a population-based genetic distance, so looking at differences in DNA between Old World and New World plant populations of the plant Arabidopsis thaliana, which is a model system. It's a weed from the mustard family. It's not useful to us as a domesticated plant. We don't eat it, but it has traveled around the world along the timescales as humans have migrated across the world. So I was really intrigued by the idea that DNA could tell us something about historical migrations. And that's a theme that stayed with me throughout my career, as you'll see in this talk. So I entered this competition, the Science Talent Search, and I ended up placing 4th in the United States that year in 1998. So this was the year I finished high school. Here's a picture of me presenting my work at the US National Academy of Sciences. And this was a really important event in my life, being just a finalist in the Science Talent Search. There are 40 students chosen each year. We were flown to Washington, D.C. to spend a week together. And I think that event played a role in my life, like this conference might for many of you. I met people like me who were really interested in science and really not shy about that. And also the fact that all of us had been mentored at such a young age, we were very lucky, and many of us are still in touch. Of the 40 of us who were finalists, more than half now have PhDs in science, and many of us are professors. So I think those early experiences of being mentored as high school students really stayed with us and shaped our lives. Okay, so I was so I placed 4th, and I was the highest placing girl that year in the competition, and lots of fun things happened, like appearing on the Today Show. So this is the top 3 finalists and myself. We got to travel to New York and do that at the end of the week. So this was like an important kind of social and scientific event in my life. The second accident is then I went to college and I continued to study mathematics. I actually was an applied mathematics major. I took a fair number of computer science courses, but that was my first exposure to computer science was in college. And I decided to pursue a PhD in evolutionary biology sort of by accident, because the truth is that I had never taken any biology classes in college. Actually, as a child, I wasn't very interested in the natural world. I wasn't interested in birds or dinosaurs or plants. I really just liked to read books, and I liked math. I did other things. I was very into spelling bees. Some of you might have done spelling bees. I also studied Indian classical dance very seriously. This is a picture of me with my family after a performance. But I did not take biology classes. And I'll get more into why I decided to do a PhD in evolutionary biology in a little bit. But this was also an accident that I decided, I'm going to pursue this field I've taken no classes in, and also that programs decided to accept me. So why did I decide that? Well, while I was in college, I was given this book to read. And I actually ended up meeting and working with, as part of my doctorate, the person who wrote it. And this book had a huge influence on me. So this is by Luca Cavalli-Sforza, who's since passed away. And it's called Genes, Peoples, and Languages. And the introduction had a real influence on me. So this was another accident. In the introduction, Luca says that research on the evolution of natural populations is inherently multidisciplinary. And he goes on to say that research on human evolution relies on archaeology, genetics, and linguistics to give a history of the last 100,000 years when our species emerged in Africa. Increasingly, though, I would say human evolution piques the interest of researchers in anthropology, and sociology, and economics, and ecology. So we're pretty interested broadly in many different fields in the university and in research around the history of our own species. And part of what the book goes on to say is that these disciplines can be expected to converge toward a common story, and behind them must lie a single history. To some, history, including evolution, is not a science because its results cannot be replicated and thus cannot be tested by the experimental method. But studying the same phenomenon from many disciplines has the value of largely independent repetition. This makes the multidisciplinary approach indispensable. So this statement, again, had a huge influence in my career. You've all learned about science from when you were young and the scientific method, and that replicability is at the heart of science, that we have to be able to replicate results in order to learn new scientific principles about the way our natural and physical world work. But it turns out that when you study natural populations and their histories, you actually can't replicate those histories. You can try to do experiments in the lab, you can try to do experiments computationally that produce signals like what you're seeing in the natural data, but we can never truly know how a natural population evolved to be the way that it is. And so for some scientists, this seems unscientific. How can I sort of say I know the evolutionary processes that produce human genetic variation without being able to do experiments on humans? But this book gave me an idea about how one might do that. And the way is to partner with multiple disciplines. So this really excited me, the idea that the mathematics I'd been studying and the computer science that I'd been studying could be key to understanding the history of our species. And that's what led me to evolutionary biology. What I started graduate school wanting to study is how human genetic variation is geographically distributed. And so I'll start by telling you what I was working on in graduate school to set the stage for a more recent project. Okay. So at the time I was beginning graduate school, the human genome draft sequence had just been published. And one big question was, how is this single reference genome going to be relevant to all people living throughout the world? How will we learn from it? So this, trying to understand human genetic variation broadly, was my goal, to try to see how relevant that reference genome was. So we started with studies of just dispersal among humans. In organisms that have limited abilities to migrate, pairs of natural populations will show increasing genetic differences. You can think about measuring this by just looking at mismatches in DNA and computing a distance. Just like on a map, we compute a geographic distance between two locations, we can compute a genetic distance between any two organisms that have DNA. It helps if they're the same species and the genome has the same length. But this is kind of a well-known ecological and genetic phenomenon called isolation by distance. So populations that are near each other in organisms with limited dispersal will be more genetically similar than populations that are far apart. Okay. So my first graduate project was to test whether this was true in human populations. So I'm going to show you a series of graphs based on some of my graduate work. And I'll explain the x-axis in more detail in a little bit. But for now, just consider this to be geographic distance between populations that are 2-generation indigenous to the location the data was taken from. And the data here are 52 populations from throughout the world, from 6 different continents, and a variety of subsistence strategies, a variety of languages spoken, and population sizes. On the y-axis is a measure of genetic distance. Okay, so you're going to see 52 choose 2 points. This is all pairs of populations. And first I'm showing you that within within continents, which have pretty low geographic distances overall, there's very low genetic differentiation on the y-axis with some variability. If we then move to look at pairs of populations where one is in Eurasia, so Europe, Central, South Asia, and another is in East Asia, then you see a sort of slight increase in genetic distance between them. If we look at pairs where one of the populations is in Africa today and the other is in Europe or Central South Asia, we see slightly more genetic distance. And the largest genetic distances and also geographic distances are between pairs of populations where one is in East Asia and one is in Oceania or indigenous American populations. So this is the kind of pattern that looks like what's called isolation by distance. It's a stepwise accumulation in genetic distance across continental barriers. And it's actually very well predicted here just by geographic distance. So what this indicates, based on these discrete jumps, is that crossing continental boundaries was rare for ancestral humans. Once our ancestors moved into a new continental region, it was quite rare that they would sort of move back to reduce these discrete jumps in genetic distance. Okay. But we wanted to sort of go beyond this analysis. So one question is, what if we calculate the geographic distance a little bit more in a nuanced way? What if we account for the fact that our ancestors couldn't cross oceans until recently, until the development of ships and long-range travel? So if we think about what was happening, 100,000 years ago when our ancestors were emerging out of Africa, they were walking. So what we did is, into the geographic distance calculations, we introduced what were called waypoints. So the idea is that these are all the circles here are the sampling locations of the Human Genome Diversity Panel. And what you can see here are just different arrows that indicate how we might have calculated distances. So there were some distances that were forced through the northeastern tip of Africa, for example, which is in keeping with how our ancestors left Africa, because crossing the Mediterranean was very difficult. And similarly, individuals ancestrally who went from Asia to the Americas crossed the Bering Strait in order to do so. So they used a land bridge that emerged when the oceans were receding. So we just incorporated about 5 of these waypoints to calculate some of these distances. Then we identified a very distinct human genetic signature, and that is decreasing genetic variation within populations with geographic distance from Africa. I'll show you what this result looks like now. So here on the y-axis, now I just have 52 points. I'm computing genetic differentiation within populations. And I'm computing this as a measure of the number of heterozygotes at different markers throughout the genome, averaged over all markers. And this is assuming Hardy-Weinberg equilibrium, if you've learned about that concept in biology, but you can just think of it as measuring the number of heterozygotes in a population. And on the x-axis is geographic distance, computed using these waypoints. So what do the colors represent? These represent the continental origin of the different individuals who were sampled. What you can see is the most genetic diversity exists in populations that live in Africa today. And that's in keeping with the fact that our species originated from Africa. That's where the most human genetic differentiation and variation still exists today. And it's a remnant of the origin of our species. There's slightly less variation in Europe and Central Asia, slightly less in East Asia, and then the least amount is observed in Indigenous American populations. And again, this x-axis, which is these geographic distances computed using waypoints, you can think of as differentiation to the origin of our species. Okay, so this is what the data show, but what's a model for how this could have happened? So next in my graduate work, I proposed a model for this. And this is called the serial founder effect model. This is a model that reliably predicts the signature of a reduction in genetic diversity with distance from the origin of the migration. So I'll show you what this model looks like in a cartoon. So imagine we have a population, and these different colored squares represent just different levels of genetic variation, just variation in DNA base pairs that exist in this population. So there's some variation. And now let's say this population becomes resource limited. People have eaten all the food that are near them. They have to move to a new place to have more resources, more animals to hunt, more food to eat, more places to cultivate food. So people leave. So maybe not everybody leaves. A small number of people leave. They take their genes with them. They take their DNA with them. They found a new population, and that population grows. And maybe the same thing happens. Resources become limited, and this process repeats itself and repeats itself. This is a fairly simple cartoon, but there are a couple of things that have happened in the course of this migration. One is that every time a new population is founded, there's a subsampling of genetic variation that occurs in each step here in this process. So we don't capture all the variation in population 1 when we found population 2, and so on. And each step is reducing genetic variation. Okay, so this is, in fact, what we think has happened in the human expansion out of Africa. And it turns out that the only genetic process that introduces new variation besides long-range migrations, which are challenging across continents, is mutation. But mutation is actually a pretty weak force in the genome in that the rates are very small. So even with a pretty not as sharp. These are called bottlenecks or founder effects. Even if this reduction in population at the founding event isn't small, it's still very hard for mutation to overcome this. Okay. And you can see this pattern of reduction in genetic diversity with distance from Africa recapitulated in many, many different ways using many different kinds of DNA markers. So even if you just look at the number of alleles at a multi-allelic locus, you see the same exact pattern that we saw with heterozygosity. And so this work was in 2005. It was published when I was in the middle of my graduate career. And since then, it's been one of the most robust results in human genetic variation. So every study since has recapitulated this trend. Some of these I was involved with, others I was not. So one study sampled many, many more populations in the Americas and found my original data plotted here. They were able to replicate that result, but then show that Indigenous American populations with much more sampling in Meso and South America have even less within population diversity than these other populations that were already studied in my work. The first studies that use genome-wide data, so this is from approximately 1 million markers in the human genome, showed the same kind of result with a decrease in diversity with distance from Africa. And this same pattern has been observed in whole genome data as well. So this pattern that we think could only be predicted by a serial founder model is observed over and over again with human genetic variation. There's another interesting pattern that emerges with newer data, especially whole genome data, which is an increase in the length of shared DNA sequences with geographic distance from Africa. This was first demonstrated, again, using genome-wide SNP genotyping platforms, which were only available about 7 years after the human genome sequence was first developed. So I know these are kind of complex terms here, but this is basically a measure of correlation or the ability to predict what I'm going to observe in the genome, given what I see at one site, what I'll see 10,000 base pairs downstream. And what this is showing is that I'm least able to do that prediction in African populations because recombination has had time to break up those genetic associations, but there are long shared sequences in Oceanic and Indigenous American populations that, again, reduce their genetic diversity. So there are lots of different types of signatures that a serial founder effect model produces, and they're all observed in the human genome. This kind of a migration history also leaves a signature on cultural traits, and that's been something else that I've studied. So when we think of genetics, we know we inherit our genes from our biological parents, but we also inherit from the society that we grow up in, the family that we grow up in, culture. That's another set of traits like language that are passed down from the previous generation to the next generation. Slightly different from genes, but with some similar characteristics. And this idea that cultural traits might parallel genetic evolution was actually proposed by Darwin in The Origin of Species. He says, a genealogical arrangement of the races of man, this is his wording, would afford the best classification of the various languages now spoken throughout the world. So the idea here is that language is important for communication. It's important for forming social groups and families that we want to, coexist with people we can communicate with, right? And that the idea here is that a genetic phylogeny or tree of individuals would also recapitulate relationships among languages. So in fact, I've done some work to test this hypothesis in collaboration with many people. In 2015, I helped co-design and lead a paper that looked at axes of variation in both genetic and linguistic data. I'll define what a phoneme is in a little bit, but that's a unit of linguistic variation. And what you can see is just that these two types of arrows in almost every continent correspond with each other. So the axes of the greatest linguistic and genetic differentiation correspond within continents. And we found that close language pairs share more features than distant language pairs. So we saw a sort of isolation by distance effect in languages, but it was a little bit different than genes in one specific way, which is that phoneme variation And phonemes are units of sound that distinguish meaning among words. So in English, for example, p and ba, the sounds that P and B make are phonemes because the words pit and bit are different words. In my mother tongue, there are other phonemes we don't have in English, like a dental D and a palatal D. So these differentiate the words kade, which means tree or forest, and kade, which means ear. So phoneme variation broadly parallels genetic variation, we found, except in isolated populations. So isolation from a genetic perspective, all it does, and you may have learned about this as genetic drift, all it does is reduce genetic variation. If a group is isolated, they just keep subsampling their own genetic variation, and actually randomly over time, the population will have incredibly reduced variation. It might see a higher incidence in Mendelian diseases. Isolation is not good from a genetic perspective. It exposes deleterious and recessive mutations. In languages, though, what we found is here, looking at languages with fewer neighbors on the left and languages with more neighbors on the right, we see that the number of phonemes, so just this measure of linguistic variability, is much higher, has a much higher range in languages that are isolated. And we were able to replicate this in a couple of data sets at different geographic ranges. So basically, all I want you to know is that repeatedly we saw isolated languages can actually have much more linguistic diversity than languages with lots of neighbors. So this is kind of an interesting counterpoint to genetics. And I think it's neat to think that culturally, isolation can actually provide the opportunity for innovation. And it seems to with respect to language. So the largest phoneme inventories occur in geographically isolated languages. The numbers of distinct and private phonemes are much higher in geographically isolated populations. One idea here is maybe that when a group is isolated, they have to innovate to keep their cultural traits kind of diverse, and they have very tight social networks. And so to describe a new concept, to describe some kind of innovation that's observed by the group but not taught by an external set of migrants, new forms of language, new words have to be developed. And these languages are also not constrained by having lots of neighbors where maybe there are people who speak dialects or they speak multiple languages in order to communicate with neighboring groups, because they're not sort of getting that influx from nearby populations. Then that leads to more innovation. So this is in sharp contrast to what happens to populations genetically. So that's some of the work that I thought about and started during graduate school. The language projects finished much later when I was a faculty member. So this is me with Mark again, popping a bottle of champagne after I finished my PhD with some friends in the background. And when I finished my dissertation, the kind of view and argument that I presented in my field, which was somewhat new at the time and is still kind of my stance in my discipline, is that neutral histories, so not selection, but just demographic changes, population founding events, particularly bottlenecks, strongly influence the fate of alleles in human populations. And I started my lab at Brown. And what I thought I'd do is to do projects like the ones I'd done in graduate school. I was going to infer detailed human population histories from diverse genomes. I thought maybe I would collaborate with anthropologists and add to the diversity of our genetic data sets. And that was the sort of view I began my lab with. But then another accident happened, which is that the cost of sequencing a human genome became really, really inexpensive relative to what it had been very quickly. So this is a graph from the National Institutes of Health of the trajectory of the cost of sequencing a human genome since 2001, when that draft sequence was published and I was applying to graduate school and decided to become a biologist, to almost today. So here's what this looks like. You may know about Moore's law, the idea that the number of microprocessors on a chip can double every 10 years, and this vastly outpaces Moore's law. So just to cast this in relief, when I was finishing college, it was estimated it cost $61.4 million to sequence a human genome. I started my faculty career at 2010, the second number, it was $31,000 to sequence a human genome. And now we're actually at numbers less than $500, which was thought to be the goal ultimately, the $1,000 genome when the human genome was originally sequenced. So what happened here is that human genetics stopped becoming data limited, but became limited by people who could develop methods and implement methods to make sense of these data sets. And how have data sets grown in my field in this time period? Just to give you a sense, some of all the data I showed you up until now were a type of marker called a microsatellite. They weren't single DNA base pairs. And I was doing that work with about 600 of those in about 1,000 individuals. So pretty quickly, once we were able to genotype genome-wide SNPs, or individual DNA base pairs, the sample sizes became about 650,000 different markers in about 1,000 individuals in 2008. Then, samples got bigger, 3,000 individuals, 500,000 locations in the genome. In 2015, a large data set called 1,000 Genomes ended up essentially sequencing and statistically identifying 84.7 million DNA nucleotides in about 2,000 individuals, 2,500 individuals. And now a data set we routinely work with is called the UK Biobank. It's genetic, biomarker, and medical records from 500,000 residents of the UK, with over almost 2 million positions in the genome assayed. So the size of these data sets and the ability to store them, the ability to compute on them has changed dramatically in my career. And now I want to transition to the last part of the talk and telling you about what these data sets allow us to understand about biology that we couldn't understand from a more hypothesis-driven standpoint. So I'm going to tell you about a paper that came out a few years ago from my group. It's led by one of my former graduate students, Priyanka Naka, who's now a senior scientist at Regeneron, and a graduate student, Sam Smith, who has just graduated and is now a postdoc at the University of Texas, Austin. And it was a collaboration with 23andMe Incorporated. Okay, so another consequence of the serial founder effect out of Africa is that there are populations. Well, every population has individuals that have long runs of homozygosity, where they have the exact same sequence of DNA inherited from both parents. And these, what are called ROH, increase with genetic distance from Africa. I'll show you what this looks like. So an ROH is a long homozygous genomic region in an individual. It ranges from about 800 base pairs, or 0.8 kilobases, to about 10 million base pairs in globally distributed human genetic data sets. So some of these can be, quite long. And there was a study that examined their distributions in large data sets, some of which I've already talked to you about, using about 500,000 DNA base pairs, or SNPs. Okay, so here's what this graph looks like. On the x-axis, every location is just a population. They're grouped by continent of origin. And these are called violin plots. You might not have seen them before, but they're just like box plots. They're just slightly different. They show the sort of shape of the distribution and the median or mean. And this is just the total length and runs of the homozygosity across all individuals in these populations. So you can see that, the total length in megabases, the human genome is about 3.2 billion base pairs. So it's fairly small in African populations, but it gets larger and larger with distance from Africa, much like things I've shown you already in this talk. And over here, these gray populations are populations of mixed ancestry. So these are African-American individuals from the US Southwest, and these are Mexican individuals living in LA. So they have inherited genetic, their DNA comes from different ancestral groups. And so that's why their runs of homozygosity sort of don't adhere to this pattern, because they have sort of complex ancestry. Okay, so this is sort of overall, you get more homozygosity with distance from Africa. But there are these really long runs of homozygosity, so about 800,000 base pairs, that correspond to isolated populations or populations that practice close kin marriage. So here, the higher means and larger variances, so if you see these stretched out violin plots, they occur in Middle Eastern groups that practice what's called endogamy, close kin marriage culture, also Central and South Asian groups that do the same. And then Indigenous American groups have been fairly isolated. So that's part of what's reflected here. And one reason this is interesting is because runs of homozygosity are thought to be deleterious because, again, recessive mutations are inherited in 2 copies. In a long run of homozygosity, that could mean producing a deleterious or a disease phenotype, okay, like we might learn about in genetics in school, things like Tay-Sachs disease, other Mendelian disorders. So when collaborating with 23andMe, we started a project. Priya was interested in doing an internship, and the question we were focused on is, how are runs of homozygosity distributed in a human genome? How do we understand where these patterns are coming from? And just to kind of disclose what was happening here financially, Priya finished her PhD in my lab, but she was a paid intern at 23andMe, which was a really great professional experience for her as part of this collaboration. And she was partially funded by 23andMe. But 23andMe did not fund me directly, but they did obviously support this research. Okay, so just to tell you about 23andMe then, some of these numbers, I'm sure they're bigger now. And many of you may have done the kit. I have myself. There are over 5 million customers in the data set. That's grown quite a bit. Many of those customers consent to research using surveys. They answer questions about their own medical history. And thus, there are about 1 billion plus phenotypic data points collected through surveys. Okay. So as we were doing this project, there was an interesting result that actually a patient posted about on Reddit, and I'll tell you about that now. So many individuals, once they do the 23andMe kit, might buy a kit for another relative in their family. And so there are lots of parents and children, and sometimes even grandparents in the data set. So parent-child data from 23andMe has actually shown that there can be extremely long segments on individuals' chromosomes, extremely long runs of homozygosity. So in parent-child duos, genetic segments should be inherited identical by descent from parents. So that means it should be clear if a parent and child are in the database, you can actually infer what DNA that child has inherited from that parent. And those genomic segments, which are called identical by descent, are reported to customers. And in fact, these can be used to find relatives for adoptees or to find relatives in the data set. Okay, so here's what this customer posted on Reddit. So these are chromosome numbers here, chromosomes 11 through 15. And this individual and this individual's father were both in the 23andMe database. So 23andMe is reporting, okay, you 2 people, child and father, are half identical on these chromosomes, which is as it should be. A child and their biological parent will be half identical on all their chromosomes. But then on chromosome 16, there's this sort of completely gray result reported, and this individual finds out that they are not identical at all to their father on that chromosome, but they're half identical on all these other chromosomes. So this isn't a question of whether this person knows their parent. It's that their parentage on this one chromosome is not represented. Okay, so this is a very weird pattern that I actually didn't know about until we observed it in the data set. But it's actually a well-known medical phenomenon called uniparental disomy. So we learn that, as I've said already in this talk, we each get half of our DNA from our biological mother and our biological father. But now I'm actually going to tell you about individuals for whom that's not the case. So they have UPD. UPD, or uniparental disomy, is the inheritance of both homologs of a chromosome from one parent with no representative copy from the other parent. So again, this is not an issue of not knowing who one's biological parent is, because it usually only happens on 1, maybe 2 chromosomes. It shouldn't happen. If it happens genome-wide, that's a sort of difference in maybe a biological relative versus a familial kin. So this is sort of a chromosome level signature, but not a genome level signature. Okay, so how could this happen? This happens usually due to errors in meiosis, so the formation of gametes. In normal meiosis, there's crossing over that happens in prophase 1, and then the homologous chromosomes, they swap material, and then they separate, and the sister chromatids separate further in meiosis 2, and there are 4 haploid gametes that go on to fuse if a zygote is formed with another gamete from another parent. This is proper meiosis. So we get a disomic zygote, and the representation from both parents is some kind of potential mosaic of the 4 grandparents. Okay. Here's an example of what happens in UPD. In UPD, there's an error that might happen during meiosis that produces, say, disomic gametes, so gametes in which non-disjunction has happened. And maybe you end up with a trisomic zygote. But instead of that zygote just stopping development, there is actually a process that we know very little about in cell biology, but by which one of these 3 chromosomes, where there should only be 2, gets degraded and kind of ejected from the cell or absorbed within the cell, such that the right number of chromosomes is results. So I'll explain different types of UPD in a minute, but this is an example of how this might happen. Okay. And this is thought to be deleterious, again, because there are clinical consequences that happen when chromosomes contain genes that are expressed in a parent-specific way, and when recessive phenotypes are unmasked by homozygosity. So UPD is known, but it presents usually because someone has a disease, what's called often an imprinting disorder, and they come to a clinic with symptoms and are then diagnosed as having a certain disorder that is a consequence of UPD. Okay. So there are many clinical UPD cases that have been known about. The first case was documented in 1987, and now there are about 3,300 clinical cases in the literature. And usually these present in very young children, so they're identified pretty early. This is from a database online about UPD in the clinic. So chromosomal rates of UPD have been estimated. With genome sequencing, we can even discover whether the UPD came from the father, or paternal UPD here, or from the mother, shown in blue, maternal UPD. So you can see that maternal UPD is more common than paternal UPD. There are many cases observed on different chromosomes that contain these imprinted genes where the parent from whom you inherit the gene is important. So that's where the clinical cases tend to be enriched. But there are many open questions about UPD. So first, we don't really know how common UPD is in the general population. The rates have been estimated from clinical cases, but not by taking a random sample. Not that 23andMe is a random sample, but we don't know the rates. We also don't really know the rates of maternal versus paternal UPD and different types of UPD that I'll define in a minute. And we don't know how it happens. The idea that errors in non-disjunction could be corrected was unknown until recently, and we don't know the mechanism by which that happens. So it's a rare phenomenon, and this is why having lots and lots of data can help us understand it better. Okay, so there are 3 subtypes of UPD. There is what's called heterodisomy. So here, non-disjunction happens in meiosis 1, but it produces kind of in the end, the one parent is not represented on a chromosome, but instead 2 grandparents from one side are represented. So here we might not worry about clinical consequences. We call this heterodisomy. There are still normal levels of genetic variation, but instead of having representation from 4 grandparents on 2 chromosomes, we only have representation from 2 grandparents on 2 chromosomes. We can also have non-disjunction in meiosis 2, in which case we have totally homozygous chromosomes, and this is called isodisomy. This is thought to be the most deleterious form of UPD. And then we can have something in between called partial isodisomy. So I'm going to have these 3 glyphs to represent these 3 different situations, but basically the point is that in the middle one, you have complete homozygosity on an entire chromosome. And in the third one, you have lots of homozygosity, but not the full chromosome. So how do we detect UPD computationally? So these are the different sources of these errors, non-disjunction in different parts of meiosis, and we want to detect it with runs of homozygosity. So in that case, we're looking for really large blocks of homozygosity in individuals with isodisomy and maybe those with partial iso-UPD. And if we have parent-child data, then we can also identify any of these using identity by descent segments, like that customer who reported this result that was a UPD result. Okay, so we applied an algorithm using parent-child pairs to identify UPD in the 23andMe database. We used almost 100,000 parent-child pairs, and we found almost 200 individuals with UPD. So now this is our estimate of rates of UPD, maternal in blue again, and paternal in red across the genome. We observed no cases on chromosome 18, but we observed cases everywhere else. We find that maternal UPD is even more common than predicted from clinical cases than paternal UPD. We see high rates of it on a variety of chromosomes. And our estimates from clinical cases were that the rate of UPD was 1 in 3,500, maybe 1 in 5,000. But our estimate is 1 in 2,000 using these trios. So we think that UPD is twice as common as was originally thought, which is a pretty new result. We also find that clinical UPD cases are biased towards chromosomes that produce imprinting disorders. So here I'm showing you just a bivariate plot. On the y-axis are our estimated UPD rates. On the x-axis are the clinical data. And the point here is just that there's no correlation here. So we're just observing a very different set of UPD cases than are in the clinical data, suggesting again that individuals can have UPD but not have any clinical consequence, which means we don't fully understand how UPD happens. Okay, since I'm running out of time, I want to set up the problem we were trying to computationally address quickly, but then I'll wrap up. So we want to use runs of homozygosity to detect UPD cases. So the idea here is I'm going to show you pictures of chromosomes with runs of homozygosity identified in them. And this you might not be able to see at all, and that's by intention. So here I'm showing these really long runs of homozygosity in green. And this is what a normal non-UPD individual looks like. You might not even see any green here. There are just 3 or 4 spots with a long run of homozygosity, which is more than 1 million base pairs. So there aren't many of those here. This is a standard genome. This is what an individual with heterodisomy looks like. Again, this individual just has grandparental representation from one side of their family, but no sort of obvious change in genetic variation. This is what partial isodisomy looks like. So on chromosome 1, this individual has really long stretches of homozygosity. And here's what isodisomy looks like on chromosome 1. So we found individuals like this in the data set. And again, they report no clinical consequences. Okay. So here's what different subtypes of UPD look like on the right and a control individual on the left. So this is kind of the machine learning problem we wanted to implement. Can we distinguish the 2 on the right from the 1 on the left? And maybe we can't distinguish heterodisomy at all. So what's the challenge here? Maybe if you look at this, you say, okay, I'm going to cover up the second picture, but I could train an algorithm, or I imagine with my eyes that I could distinguish the 2 rightmost plots from the leftmost plot. The problem is that if individuals have related parents, which doesn't mean closely related, even if they're distantly related, then you get long runs of homozygosity all over the genome. So here, this is UPD of chromosome 1 in ground truth, but these individuals also have long runs of homozygosity in other parts of the genome. So would we be able to classify this properly? And even the control individual who does not have UPD also has lots of long runs of homozygosity, again, because of the serial founder effect out of Africa, ROH varies by chromosome and ancestry. Okay, so in order to do this, we built a machine learning classifier. It took into account, I'm going to skip this very quickly, total ROH, different proportions of it, the maximum length on a chromosome, and the second largest length. And then we classified different individuals in the database as likely UPD candidates versus others. So now we find in over 1 million individuals in the database with no relatives, over 304 putative UPD cases. We can't decide whether these are maternal or paternal because we don't have parent data for these, but the chromosome distribution looks very similar to that with duos. We replicated this. This is what my student Sam did, who was second author in the UK Biobank, and we identify about 172 UPD cases in that data set. And coming back to the original question, we are able to study the distribution of homozygosity runs in individuals that are putatively healthy. We can detect this phenomenon, UPD, computationally at scales that were not possible before. So before it was detected one by one in the clinic, but not in a kind of data-driven way. We estimate an overall rate of UPD that was unestimated before, and that it's twice as common as thought from clinical cases. So now what we're working on since this paper is to identify whether these individuals are deficient in recessive mutations, which appears to be the case because they don't report deleterious phenotypes in these data sets. And we think that's because the individuals we're observing are live-born, they're adults. So, they don't have any deleterious mutations. If they did, that would have been, there would probably be embryonic selection against that. And so this to me is exciting because it shows that you can have long runs of homozygosity and actually not have clinical consequences of that. Okay, so when we published this, there was a little bit of Twitter backlash, but that was partly from people not understanding the title. So this was a nice amplifying tweet someone made about the importance of the word prevalence. But just to revisit where we began. So the serial founder effect model is really interesting because it says that geographic distance can predict genetic distance broadly. And it actually tells us that the effect of natural selection on human genomes is pretty low compared to the effect of our population's histories. So now when I think about different accidents that have shaped my career, I think about all the people who've come to work with me in my lab, who've been interested in these kinds of questions, who are interested in studying the role of history versus selection in shaping human genomes. And we study the shared history of our species, how we need to condition on it to identify adaptive mutations, and how mutations interact to produce different diseases in the human genome. And I know I've thrown a lot at you today, but what I want to close with is by saying when I look back on my career, I think of it as one where I'm interested in our genome as a historical text. We talk a lot about our DNA being really important to us. This is a sign in Sweden during the Winter Olympics saying the winter is our DNA. Now we're going to show it. I think the idea that our DNA kind of resonates with us culturally and as scientists is interesting and important to talk about, and that we should use it to highlight the biological nature of our shared humanity. And that's something I write a lot about too. So just to close with you, when I look back at this picture now from 24 years ago, I think about the accidents that have shaped my career, many of which I've told you about. But then I think about the fact that I'm still working on the same questions that I was captivated by as a 15-year-old, and suddenly my career feels like not an accident at all. So thank you very much, and I hope you enjoy the rest of the conference.
Thank you so much for a wonderful talk. A friendly reminder to everyone, make sure that you are putting your questions in the Q&A. Does anyone have any questions? I know Darsh is planning on coming in and may have a couple questions for you as well.
I'm here. I'm monitoring the Q&A chat, so I will let you know if something pops up.
All right. I think we only have about 1 more minute, and then we will have to slide into, well, hold on. We have a few more minutes, I guess.
I know, it went a little long.
I know. No, you're totally fine. You're totally fine. That's exciting though. When there's something exciting to talk about, you get to go on about it. So it's good. We have about Actually, we have a little bit of time. We have about 14 more minutes. So if anyone has any questions, Darsh, if you or Toba have any specific questions, please feel free to ask them now.
Yeah, it looks like there is one question in the Q&A session.
Great.
So this is coming from Nasera. She's asking, could you speak more on the cultural or ethical aspects of your work?
Yeah. Thank you so much. So for sure, this kind of work requires, again, I think, an interdisciplinary approach. So a lot of collaboration with anthropologists, many of whom are gathering the data and doing community engagement with many of the people that are sampled in these studies to communicate results back to them, to again highlight that when we study human genetics, we're studying how similar we all are. So that I came in at the very end of the talk, but just for some numbers for all of you. So our genome is about 3.2 billion base pairs of DNA letters long, but any two of us only differ in 1 in 1,000 of those base pairs. We're incredibly similar. And that, I think, is what is really exciting to me about genetics, because when the Human Genome Project started, the promise was that the genome by now, or for maybe my children, for all of you, will be integrated into personalized medicine, that, the way that we are treated in our doctor's offices, the therapies and the lifestyle changes that are recommended to us will take our genome into account. And I think that the most exciting thing that we've learned in the last 20 years is that our genome is, in fact, even though it resonates with us and people will say things like, oh, enjoying math is in my DNA. The truth is our genome is not a blueprint, right? Our outcomes in life, the things that we do, the diseases we get are not largely predetermined, especially, after our early childhood years. And so I think what's really exciting to me about this work is to highlight the importance of our environment, to highlight the, the important role we play and our social networks, our families play in building our environments and advocating for others in our environments. And in highlighting that, again, a single reference genome is not actually going to help us understand human similarity. And, my children are biracial. It's not, a genome that doesn't represent them is not going to help them get better healthcare as they get older. So I would say I'm very motivated by the cultural and ethical aspects of genetic research, and I think a lot of my colleagues are. That said, so I'll say first, community engagement with groups that are contributing DNA is incredibly important, and it's something that I have many colleagues who work really, really hard on, and I'm really grateful to them, because without them, like, my computational analyses would not be possible. But ethically, I think there's also, increasingly really important reasons to be interested in human genetics. And I actually recently have a piece that's co-authored that's in Nature on this. They had a special issue in October on racism in science. And some of you might be interested to read this piece. It's about the fact that in the United States and actually throughout the world, extremists use genetics and have historically. Genetics and extremist political values have kind of been intertwined. So some people who want to claim superiority over other people try to use genetics to justify that, and that's not justified at all by genetics research. And I think that's a really important thing for scientists like myself to say, and to say loudly. And again, to say that, what our research shows is that there isn't a biological basis for any group to consider itself superior to another, and also that what biology shows is, again, how similar we all are, and that we should, use it to improve lives, not to discriminate. And so that is another, I think, important feature of being a geneticist, is engaging in that social conversation about how this work is relevant to all of us and relevant to how we create just societies.
Awesome, thanks. There's one more question in the Q&A chat. I hope I'm pronouncing their name right. This is coming from Kasadech. The question is, with the current progress of technologies, how would you picture the direction of your work in this particular field?
Yeah. So I think that is a really great question. I think an interesting feature of a scientific career, especially a career as a professor, is it's very much a career that shifts at some point to being about mentoring young scientists, which is what I love about it, actually. I mean, one of my goals in my research life is to mentor a young scientist to do a high school science fair project, much like I did to start my career. So I guess I say that to say that this is a great question, because the reality of being a professor is that at some point, and for me, it's kind of happened after a decade of being a professor, but sort of 20 years out from starting graduate school, the technologies we use in my lab today are so far beyond what I thought would be available when I was a graduate student. I'm no longer kind of well versed in them. My students are. The people who work with me, undergraduates even. But I'll say, in terms of technologies, there are two things that I think have really changed the field. One is our ability to amplify DNA from bones. So this is the basis of the Nobel Prize in Physiology and Medicine this year that went to Svante Pääbo. So he developed the ability to extract DNA first from a mummy and then from bones to study, the Neanderthal genome, but also increasingly DNA from populations using archaeological data. And so this lets us study human evolution and kind of calibrate it at different time points. However, this has its own cultural and ethical challenges. Obviously, excavating human archaeological sites has a whole host of ethical challenges. Interpreting the context of those is a really interdisciplinary effort. But also, ancient DNA in bones, is only really well preserved in cold climates. So in fact, we know very little still about the origin of humans, because ancient DNA in Africa, it kind of is incredibly difficult to recover, and certainly not on the timescale of the origin of our species. So that's one kind of data that I think is really exciting, and we've started to work with it. I have colleagues who have done a lot of work on it, like Amelia Huerta Sanchez, my colleague here at Brown. But I wouldn't say it's sort of revolutionized my own work, but it certainly is a new line of data that we didn't have and that we're continuing to start to use. Another is CRISPR technologies, which are still hard to use for functional validation in humans. So when we study things like the genetic basis of disease, there are individuals who will then CRISPR that into an immortalized cell line and try to study consequences. But I think for me, actually, what's changing is I'm getting more and more interested in studying non-human organisms. So organisms that we've domesticated, like canids, dogs and wolves. Also mosquitoes that are vectors for diseases like malaria, chikungunya. And I think that in those systems, using CRISPR can actually let us understand how to curb disease or how to study selected phenotypes, and the basis of natural selection. So that's, I think, what's really changing for me, is both expanding beyond humans and thinking about experimental validation, and also ancient DNA.
We have one more question in the Q&A. Daniel, the question is, could you explain more on how we can use genome sequencing to prevent and treat infectious diseases and cancerous disease?
Yeah, this is a really great question. So with infectious disease, I think that sequencing has to be part of a global and local response to infectious disease. And I worry that with the COVID-19 pandemic, I'm not sure how much we learned, like how to sort of implement this pipeline when something similar happens in the future. But I don't know how many of you have looked at, there's a website by a US scientist named Trevor Bedford called nextstrain.org, NEXTSTRAIN, nextstrain.org. And he was uploading and visualizing data from COVID-19 tests and sequences gathered from the beginning of the pandemic, and constructing phylogenies to show the spread of the virus. And this, was really fascinating. You could see it in March 2020 arriving throughout the world based on flight patterns. I saw on the website how it entered my home state of Rhode Island in the US, because people were traveling during school breaks. to different parts of Europe and came in contact with people who had traveled from China, and that's how it entered our state. So I think that, sequencing can be part of tracking, but it can't really solve the need for a quick epidemiological response, and things like shutdowns and vaccine dissemination are part of that. I think that in terms of cancer, genome sequencing is hugely important. It's important in two ways. One is to sequence tumors and try to understand what the drivers of the tumor proliferation are, what is actually disrupting proper cell division. But another is to actually use our inherited genome, what we call our germline genome, what I've been talking to you about in this talk. How do we use that to try to predict both response to cancer therapies and also predilection to cancer? And so, I think genomics is key to all of these things. And we've done some work on that, both in, response to therapy in childhood leukemias, and also in studying efficacy of immunotherapies in cancer. So, I think the genome holds a lot of secrets, and it's kind of a exciting book for us all to read together.
Looks like we have one more question, and I think this is probably going to be our last question. It's so Nasra's fine question on UPD, and she's wondering how it's detected.
Yeah. So the way that we can kind of gold standard detect it is if you have a child and data from one of their biological parents, then you can detect whether there's any chromosome in their genome where large parts of the chromosome do not represent that parent. If you have both parents, you can, also detect it. In that case, if you have parent-child data, you can even detect that first subtype I talked about, heterodysomy. So often what happens in the process of clinical UPD diagnoses is a child presents with a series of symptoms, that maybe puzzle a few doctors. And then eventually someone says, oh, we should do some sequencing. They sequence the child's genome, and if they can sequence both parents, they do that as well. And then they compare them and they identify it. What we were trying to do was develop a method using runs of homozygosity to identify UPD without the need for parental DNA. But then you're restricted to only being able to identify a couple subtypes that are characterized by really long blocks of homozygosity. And the specific pattern one is looking for is with parent-child data, single chromosomes that show no representation of that parent or of the other parent, which again, if you have one parent, you can kind of paint the chromosomes as this came from the other parent, even though I don't have their DNA, and this came from this parent. We did, in some of our runs, identify individuals who had UPD on 2 chromosomes, but we didn't see anybody with 3, and I think that would be pretty unlikely in a live-born individual. So I hope that clarifies it. I know I went over it quickly, so thank you for asking.
Awesome, thank you so much. I actually had one quick question myself.
Mm-hmm.
Going back to the geographical linguistics thing, do we see? I'm not sure if I missed this or, but I just wanted to ask real quick, like, do we see phonemic similarities between languages that are that might be unrelated but still are in close geographical proximity?
Yes, we do.
Okay.
So so we see more phonemic similarity between geographic neighbors than we do from distant
Okay,
geographic.
interesting. Okay, awesome.